Okay, so what we're going to do today. Last time we gotten as far as describing the components of DNA. Today what we're going to do is extend that story into the components and talk about the fact that DNA can replicate itself. Again, it's really hard to overemphasize the importance of DNA. An understanding of DNA over the last 70 years has allowed us to basically revamp biology. And so because DNA is the molecule where we store all of the information inside our cells, in prokaryotes and in eukaryotes, about the kinds of proteins that can be made, it's an essential molecule for life. But not only is it a kind of storehouse for information, not only does it provide this kind of blueprint, but in addition, it has the ability to replicate. It can make copies of itself. And one of the things we're going to talk about today is how this molecule has the capacity to replicate itself. Now, it requires the help of a lot of other proteins, and we're going to just talk about a few of them. If you take another cell biology course, you'll find there are a lot of details I'm leaving out. But nonetheless, we'll get the basics about how we can replicate a molecule of DNA and make new copies of the DNA. So again, to be considered life, the life has to be able to replicate itself. And so we'll see that DNA follows that requirement. It can replicate itself, and also to be an aspect of life, it has to change over time. And it will change over time because of mutations. So that then provides the basis of evolution. But we'll talk about this briefly. The components are basically, there are 5 components of DNA. There's a 5-carbon sugar, deoxyribose. There are the 4 nitrogenous bases, adenine, guanine, both of which are purines, and another 2 called cytosine and thymine, both of which are pyrimidines. They have slightly different shapes. And one of the insights of Watson and Crick in the early 1950s was to recognize how these molecules could fit together, how they could mechanically interact with one another. Later we realized this was through hydrogen bonding, and I'll tell you a little bit about that story. The next thing we're going to do is we're going to take these components, the sugar,
You want to try talking?
Okay, do we have sound? All right, so where did I leave off? Shall I start from the beginning?
No, just backtrack.
Okay, so we've gotten through this idea of the components of DNA being deoxyribose and the 4 nitrogenous bases broken up into the 2 purines, adenine and guanine, and the 2 pyrimidines, cytosine and thymine. Then what we want to talk about is the idea of the organization of the 2 strands. And we'll talk about this in terms of understanding how this molecule can replicate itself. The importance of a molecule replicating itself is the basis of life. So we're going to see that it's this molecule, the molecule of DNA, which stores all the information for the amino acid sequence in all proteins. Again, this will be back to the central dogma. DNA gives rise through transcription to RNA. RNA gives rise through translation to protein. And that's basically what we're going to be talking about for most of the rest of the week here. And what we've gotten to here is the idea that the DNA molecule is going to be composed of two strands. And the two strands are going to be held together by hydrogen bonding. So let's look and see how we're going to construct that molecule. Each of the two strands is made up of a backbone. Each of the two strands is, in fact, a backbone constructed of deoxyribose. Each of the deoxyribose molecules is linked to its neighbor through a particular kind of covalent bond called a phosphodiester linkage. We'll see how those phosphodiester linkages are made, and we'll see that there's always this particular fashion in which these deoxyriboses are linked one to the other. They're always linked between the number 3 carbon and the number 5 carbon, and we'll talk a little bit about how that happens. But the backbone is constructed of the deoxyriboses that are linked together by phosphodester bonds into this great long polymer. And then we'll see that there are only two ways in which we can hook up the bases. The bases have to be arranged adenine to thymine or guanine to cytosine. You notice the way I've paired these. Adenine and guanine are both purines, thymine and cytosine are both pyrimidines, so the purine adenine always base pairs with the pyrimidine thymine, and the purine guanine always base pairs with the pyrimidine cytosine. It took a long time to sort that out, and I'll tell you a little bit about how that was done. But fundamentally now what we have is we have covalent linkages that through the phosphates that are tying together all of the deoxyriboses. Then those two strands, those two backbones, each one of the deoxyriboses having attached to it a base. Those bases now, adenine to thymine, guanine to cytosine, will base pair, and they'll base pair through hydrogen bonding. So the two strands of the DNA molecule are being held together by hydrogen bonding, whereas the strand itself, composed of the backbone of deoxyribose, is held together by covalent linkages called phosphodiester bonds. One of the other characteristics of DNA that I'll try to draw on the board, and I can only do this in 2 dimensions, and even then I'm a little taxed. And what we'll see is there's a particular orientation of the two strands. One is oriented 3' to 5', and the other strand is oriented in the opposite direction, 5' to 3'. These terms, 3' to 5' and 5' to 3', are describing the positions of the carbons on the 5-carbon sugar deoxyribose. We'll see that the linkages are 3 to 5, and this now will give an endedness to the molecule, but that the two strands are pointed in opposite directions. They're said to be anti-parallel, and we'll draw that in the board and talk about it. The next idea is how we're going to take this molecule, which is constructed in this fashion, and what are the rules we're going to use to replicate the molecule, to make copies of the molecule. And when we make copies of the molecule, DNA replication is said to be semi-conservative. Semi-conservative has a real meaning here. It's not like a description of the current Republican Party. It is a description of how the template strand provides the old strand, and the new strand is now complementary to the old strand. So we'll take these two old strands and make new strands that are complementary to them. And that's what we mean by semi-conservative replication. The replication mechanism, again, we're going to boil this down to a handful of proteins. If you get into the weeds of this, there are a few more of them. There's some very interesting things about the replication. But the major players I want to talk about are first a helicase. Remember that the DNA molecule, its lowest order thermodynamic state, is such that it's thrown into this double helix. So first thing we have to do to begin replicating the molecules, we have to unwind the helix. We use a particular enzyme called a helicase. I'll show this helicase as a kind of wedge, and it's useful, I think, because we can imagine that wedge getting in between the hydrogen bonding, between the purines and pyrimidines, and splitting those hydrogen bonds, separating the two strands. But then we need another enzyme. We have another enzyme that comes along called a DNA polymerase. And again, there's meaning in this term, DNA polymerase. We're going to polymerize a new molecule of DNA. And what we're going to do is we're going to use the template strand, and we're going to walk along the template strand with this DNA polymerase. And the DNA polymerase has two roles. It first picks the appropriate base so that each base in the old strand will be complementary to the base in the new strand. So the DNA polymerase selects that base. Secondly, the DNA polymerase forms the phosphodiester linkage. It's this enzyme that forms the phosphodiester linkage between adjacent deoxyriboses. So its two roles are to select the appropriate base, and secondly, to form the backbone by forming those phosphodester bonds. There's a constraint here, though, and this constraint is that the DNA polymerase can only read in one direction. It can only read from the 3' to the 5' direction. And yet we're going to see that the two strands of DNA, two strands of, in the DNA molecule, are oriented anti-parallel to one another. So that invokes a certain problem, that the DNA strand is going to be read only from the 3' to 5' direction, and yet the two strands are oriented opposite to one another. This gives rise to the concept of a leading and lagging strand, and we'll talk about what that means. Fundamentally, what it means is the DNA polymerase can only read the substrates, the underlying strand, from its 3' to the 5' direction, and generate a new strand, 5' to 3'. When you separate the two, that means one, the leading strand can be read continuously, the lagging strand has to be read in fits and starts, and we'll talk about why that is. But again, let me remind you about the fundamentals here, and that is the structure of DNA. It's a 5-carbon sugar. And the precise structure of this molecule is fairly important. So it's going to dictate a lot of what we're going to talk about from here on for this lecture, and to some extent tomorrow and Friday. It's a 5-carbon sugar, and we can, at each of these vertices, there's a carbon. So I'm not going to draw the carbons in, but I will remind you there's a carbon here, a carbon here, a carbon here, a carbon here. But if you count them, that's 1, 2, 3, 1, 2, 3, 4. So there's a 5th carbon up here. If we number the carbons as 1, 2, 3, 4, and 5, and look at the details here, we'll find that at the number 1 carbon we have a base. It's true there'll be another hydrogen in here to make up 4 bonds. The number 2 carbon has a hydrogen and a hydrogen. This is important because when we talk about ribose, a related 5-carbon sugar, the only distinction between the deoxyribose and the ribose will be here at the number 2 carbon. As the name implies, deoxyribose lacks an oxygen here. So there are the 2 hydrogens. When we get to ribose, we'll see there's a hydrogen and a hydroxyl. Well, over here on this number 3 carbon, there's a hydroxyl and a hydrogen. In the number 4 carbon, there'll be a hydrogen and a hydrogen, or just 1 hydrogen. I'm sorry, there are 3 bonds there. So 1, 2, 3, and the 4th is a hydrogen. And up here on this carbon is a hydrogen, and a hydroxyl. That's deoxyribose. That's the 5-carbon sugar. The base out here at the number 1 carbon the bases in DNA that are covalently linked to the number 1 carbon of deoxyribose can be of 4 types. That is, they can be adenine or guanine. Again, I want you to know that these are both purines. And then the other 2 possibilities are thymine and cytosine. And these are pyrimidines. The rules are that we always bind a purine to a pyrimidine through hydrogen bonding, and I'll show you some pictures of that. But basically, we're talking about an adenine, always base pairing, to a thymine. So again, here's the purine, This is the pyrimidine. The other possibility is to use the other purine, guanine, to base pair to the other pyrimidine, cytosine. So these are the base pairs, AT, CG. And we'll see here in a little bit that these are going to be held together by hydrogen bonds. It's going to be a charge-charge interaction for these. So hold on to the idea that this ATGC bonding is hydrogen bonding. Whereas over here, what we're going to do, and we'll draw this out in a moment, is we're going to now link one deoxyribose to the next by phosphodiester bonds. These phosphodiester bonds are in fact covalent bonds. So there's going to be electron sharing here, and these are going to be very strong bonds. So these are covalent bonds, and these are hydrogen bonds. Now let's take a moment and look at how we're going to form those phosphodiester bonds and how that's going to happen. And we'll do that over here on the right-hand panel. So here's our 5-carbon sugar with a carbon up here. Let me give myself some more room. You'll see in a minute why I need more room. Here's the oxygen. Carbons 1, 2, 3, 4, and 5. Now I said up here, this carbon, the number 5 position, has a hydroxyl. Well, one of the things we can do with that is we can use that oxygen on the hydroxyl and change it a little bit. And what we're going to do is take this carbon, and now we'll use the same old oxygen as part of the hydroxyl, but now we're going to link it an oxygen to a phosphorus, to an oxygen to a phosphorus, to an oxygen to a phosphorus. And each of these phosphoruses will have a double bonded oxygen and an O negative, a double bonded oxygen and an O negative, and a double bonded oxygen and an O negative. You'll recognize this as the same sort of phosphates that we had on phospholipids. But now we've modified them a little bit, and we have 3 of them. And we're using this oxygen-phosphorus-oxygen bond. And we can look at this, and out here we can put, say, an adenine. And we can say that this is an adenine triphosphate of deoxyribose. So we've taken this deoxyribose molecule, and remember here we have the number 2 carbon, we have the 2 hydrogens. The number 3 carbon, we have an hydroxyl and hydrogen. Now, what I'm going to do is I'm going to redraw this, and I'm going to put another base in it. We could put any base, basically. The base is not important. What we're thinking about now is what's going on out here. And we're going to think about what's going on between the number 3 carbon here of this deoxyribose and the number 5 carbon of the next adjacent deoxyribose. So here we'll draw another one. 1, 2, 3, 4, and the number 5 carbon is up here. We have a base out here. It could be any of the 4 bases, but it's a base. And it too is going to have this carbon, oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. 1, 2, 3, right. Double bonded oxygen, O negative. I won't put all those in. But what we're going to find is that we're going to cleave off these outer 2 phosphates. So where this is a triphosphate, we're going to take off those 2 phosphates. And just as there's energy in ATP that we talked about the other day in terms of converting chemical energy to movement, we're now going to use the energy in this triphosphate. And with the help of an enzyme, which we'll see a little later, is the DNA polymerase. With the help of an enzyme, we're going to rearrange these electron sharing, the electron shared here. And what we're going to do is we're going to use the oxygen here on the number 3 carbon. Then we're going to have an oxygen, a phosphorus. We'll get rid of this the diphosphate. So we have an oxygen, a phosphorus, and an oxygen bond. So carbon to the oxygen to the phosphorus to the oxygen, the number 3 carbon, and this is an OPO. This is a phospho diester bond. So this oxygen, this carbon, oxygen, phosphorus, oxygen is the phosphodiester bond. We now have equal electron sharing among these phosphates and these oxygens, and we now have a covalent bond between this between this deoxyribose molecule and this deoxyribose molecule. And we can repeat this over and over and over again. So we can now take another molecule of deoxyribose. This is a base out here. It could be any one of the 4, so I'll just put a B. We have again our hydrogens. This is deoxyribose. We have again a hydrogen and a hydroxyl. Out here at the number 4 carbon, we just have a hydrogen, but the number 5 carbon now is again the important player. The carbon, oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. I won't put in all the double bonds. And again, we're going to use these 2 phosphates and the energy of these 2 phosphates, and we're going to rearrange things so that now this hydroxyl, we get rid of the hydrogen. We have an oxygen to a phosphorus to an oxygen bond. So again, this is the phosphodiester linkage. We're repeating the pattern over and over and over again. We're making a polymer of these deoxyribose sugars. We can do this down here again. I'll do it one more time. O. This is the base. It's number 1, 2, 3, 4, and 5. Again, we'll take the phosphorus, O, P, O, P, O, P. Take the diphosphate, get rid of that, and now form a new bond that goes oxygen to phosphorus to oxygen. Again, a phosphodiester bond. Now we've shown 1, 2, 3, 4 deoxyriboses, but in fact, if we think about the chromosomes in a eukaryotic organism, that's one long strand of DNA. And that means there will be literally millions of these deoxyriboses linked, each one to their neighbor, to form the backbone of the DNA molecule. Each of the deoxyriboses linked to its neighbor by this phosphodiester bond, and always, always, always oriented in such a way it goes from the number 5 carbon to the number 3 carbon. The other thing this does is the way we're forming these phosphodiester bonds is it gives a kind of endedness to the molecule. We have an endedness to the molecule such that since this is the number 5 carbon, we say this is the 5' end. And since this is the number 3 carbon, this is said to be the 3' end. So now we have this strand of DNA oriented in such a way that's a 5' end up here and a 3' end down here. What we've done so far is just lay out one strand. But remember, one of the characteristics of the DNA is that DNA is a double-stranded helix. Well, that tells us that we need to put another strand in here. And as I said in the introduction, those 2 strands will be held together by hydrogen bonds. But I also said that those 2 strands are oriented in opposite directions to one another. So now I'm going to draw the other strand, and bear with me because it's kind of upside down, and I always struggle with this. So there's the oxygen. We can number them 1, 2, 3, 4, and out here's the number 5 carbon. At the number 1 carbon here we have a base. Now since we said this is adenine, this will have to be thymine. Always adenine to thymine. Always the purine to the pyrimidine. But we can do the same thing over here, and we number these carbons. And if we look at this, let me get rid of this so I can draw this in. So this number 3 carbon has a hydroxyl and a hydrogen. The number 5 carbon is again has this oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. And we can play the same game. So now we can take another deoxyribose molecule. It's the oxygen. It's the number 1 carbon. Number them 1, 2, 3, 4, and 5. Let's say that this is a guanine. If this is a guanine, that means this has to be a cytosine. So on the number 1 carbon, we'll have a cytosine, and we'll see that we'll base pair these by hydrogen bonding. Then at the number 3 carbon, we have a hydroxyl again. And we play the same game. We take this oxygen, so this carbon, oxygen, phosphorus, and what we do is we get rid of these 2 phosphates. They're cleaved off, and we rearrange this molecule so that now we have an oxygen to a phosphorus to an oxygen bond to that carbon. And this again is the phosphodiester bond. But I think you can see what we're doing here. We're laying out the 2 strands oriented in opposite directions. Because where over here, this is the 5' end, if we count the carbons again, 1, 2, 3, this is the 3' end. And we always link the number 5 carbon to the number 3 carbon. So we do this over and over again. We'll put in another one here. If this is going to be, say, cytosine, then at the number 1 carbon here, there has to be a guanine. We'll base pair those through hydrogen bonding. But the number 3 carbon is now going to, again, be linked by an oxygen to a phosphorus, to an oxygen bond. So there's the oxygen, phosphorus, oxygen bond. It's, again, this phosphodiester bond. And down here again, we'll have another one of these guys. So if this is a guanine, this has to be a cytosine. This is the number 3 carbon, so it's going to be linked to the number 5 carbon here by an oxygen to a phosphorus to an oxygen bond. That's our phosphodiester linkage. We can do this over and over again. And now, if we look at these molecules, this is the number 5 carbon out here. So that means this strand is oriented in such a way that this is the 3' end, and this is the 5' end. This idea that the 2 strands are running more or less opposite to one another is the idea of the anti-parallel organization of the DNA molecule. Fundamentally, this is the insight that Watson and Crick had in 1953 when they sorted it out. Before that time, it was known, a guy named Chargaff, who had actually done some of the work on the Manhattan Project. Chargaff was a really good chemist. And what he had done is he had taken DNA molecules, purified the DNA, and found simply by chemistry that there was a a 1-to-1 ratio of adenine to thymine, guanine to cytosine. So for each cytosine there was guanine, for each thymine there was an adenine. So that was an indication they had to pair up in some way. If you're going to make a model of the DNA molecule, Chargaff told them that you have, for each adenine there had to be a thymine. The question at that point, though, once they started studying it, was they knew they had a deoxyribose molecule, they knew they had a polymer, they knew they had these bases. But what they didn't know is how this was going to be oriented, how it was going to be organized. And so the upshot of all of this is that Rosalind Franklin, who was a graduate student at the time with a guy named Wilkerson, and Wilkerson was an X-ray crystallographer, and she was working on the X-ray crystallography, looking at the X-ray, the crystallography structure of the DNA molecule. And Watson he was at Cambridge, and Watson then went to, I think it was London, I think it was King's College in London. He went down to London and looked at a seminar that she was giving. And when he looked at the seminar, he began to realize that she had misinterpreted that X-ray crystallography data. And there's a big controversy about her contributions to all this. We'll leave that behind. But it turned out that up to that time, people were trying to put the bases on the outside and put the sugars on the inside. But once Watson and Crick looked at that X-ray crystallography, they realized that was the other way around, that somehow they had to form these bases inward. And then what they did is they literally took cutouts of cytosine and guanine and tried to fit them together, and adenine to thymine. They took mechanical cutouts and said, how can we fit these things together? And there was only one way they could make these molecules fit together properly, and that was adenine to thymine, guanine to cytosine. Let me show you a couple of pictures here, sort of emphasize what we've been saying. The first picture is just a pretty picture of my chicken scratch on the board. But this basically is what we've been talking about. Here's the 5-carbon deoxyribose in purple. The number 1 carbon here has a base attached to it, 2, 3, 4, and 5. The number 2 carbon shows here with the red hydrogen, emphasizing that we have a distinction between ribose and deoxyribose, because here in deoxyribose we only have the hydrogen. Turns out this is important. If we put a big hydroxyl group out here, it inhibits the ability of these molecules to now turn into a helix. Then again, if we count the carbons, 1, here we have a cytosine, 2, 3, 4, 5. The number 5 carbon now is linked from a carbon to an oxygen to a phosphorus to an oxygen to this number 3 carbon. So there's the phosphodiester linkage. Again, we repeat the pattern. We link the number 3 carbon of this deoxyribose to the number 5 carbon of the next deoxyribose in the chain through this phosphodiester linkage. Again, this is a proper covalent bond. These molecules are being held together tightly by this electron sharing oxygen, phosphorus, oxygen. Oxygen, phosphorus, oxygen. We do this over and over again, always having the number 1 carbon linked to one of the bases. So they show the 4 bases here: guanine, which is a purine; cytosine, which is a pyrimidine; thymine, which is a pyrimidine; adenine, which is a purine. And so they're just showing these 4. Now, the next figure is one I like, and I'll never ask you to memorize this or any of that. But it's important to look at this for a moment, because it shows how the charge structure on the purines and pyrimidines are allowing these base pairs to form, and how this is proper hydrogen bonding and not covalent bonding. Let's take a moment to look at it. Here's thymine. Thymine has this nitrogen in its ring structure. It has a hydrogen attached to that nitrogen, and because the nitrogen is borrowing the electrons from the proton of the hydrogen, it's positively charged. Up here, the double bonded oxygen has an excess of electrons, and so this is negatively charged. So we have this negative charge on this oxygen of thymine, positive charge on this hydrogen. Now, if we look at the complementary base, now in this case, this is adenine. So this is the adenine structure. So it has this 5-member ring out here, 6-member ring here, and then there's an amine coming off of the ring. This amine, this NH2, has a positively charged hydrogen at this site. On the other hand, because of the sharing of the electrons in this 6-member ring, this nitrogen has had some of its electrons borrowed, so it's negatively charged. So that means this negative charge of the nitrogen can interact with the positive charge in the hydrogen. So these two bases, thymine and adenine, are being held together by the hydrogen bonding here and here. We can do the same pattern down here where we take cytosine, which again, remember cytosine is one of the pyrimidines. If we look at its structure, here's a double bonded oxygen, here's a nitrogen that's negatively charged, and here's an amine up here with its positively charged hydrogen. So this molecule now is base pairing with this molecule over here, the purine guanine. But again, here's that positive charge on this amine connected to the ring structure. There's a hydrogen on this nitrogen in the ring structure, which is positively charged. Meanwhile, the double bonded oxygen up here is negatively charged, and it can interact with the positive charge on this amine. So now we have a case where cytosine to guanine is bound by 3 hydrogen bonds. If you go back and look at the early literature, this when they first began studying DNA, they began to recognize that they could take DNA molecules and they could actually separate the 2 strands. And the way they could separate the 2 strands was simply to heat it. By increasing the thermal activity, they could, what they called, melt the 2 strands apart. So they would heat things up and then look at the amount of energy, amount of heat it took to melt the 2 strands. And they found that when they had strands that were predominantly cytosine to guanine, it took more energy to melt those bonds than it took to melt the adenine to thymine bonds, because there were only 2 as opposed to 3. So that gave them an indication that these cytosine to guanine hydrogen bonds were stronger than the adenine to thymine. And again, it's interesting, I think, to look at these shapes, because these are actually the shapes that Watson and Crick cut out, either of paper, and later on they had the machine shop make scale models of these molecules. And they found that there was only one way, when they actually took these scale models of the molecules, tried to bring them together, there was only one way they could fit. There was only one way the jigsaw puzzle would fit together, and this was thymine to adenine and cytosine to guanine. So that's why we always have these TA or AT or CG pairs. So any questions up to that? Okay. So what we have talked about so far are the components of the DNA molecule. We've talked about the orientation of the DNA molecule being antiparallel. But for a molecule to be the basis of life, the molecule must be able to replicate. So what I want to do on this panel is I want to talk about DNA replication. And I'm going to say first, as a fundamental idea, that DNA replication is semi-conservative. There's meaning in this term, should be an M. There's a meaning in the term semi-conservative. It means that the old strand provides a template for the new strand. Since there are always 2 strands in DNA molecule, it's probably more appropriate to say each old strand provides a template for the new strand, or the newly synthesized strand. And I'll just draw these as 2 lines and number these as 1 and 2. So this is the old strand, and this is the old strand. And semi-conservative replication means that when we replicate a new molecule, we'll have an old strand here, and in blue, we have a new strand. So this is the new strand. And I'll number these to say that this old strand is number 1. And then similarly down here, there's going to be the number 2 old strand, but it will be complementary to this new strand. And this is the old strand. So conceptually, that's what we mean by semi-conservative replication. And what I'm going to do now is I'm going to start talking about some of the enzymes that are important in going through this process and some of the tricks we have to do to make these new strands and replicate the new strand from the old strand. So any questions up to there? Okay. So if there are no questions, what I want to do here is I'm going to make a list. And I'm going to make a list of the enzymes that we're going to talk about. And again, this is not an exhaustive list. If you take another course, you'll find there's some other enzymes that are important in this process. And if you think about it for a moment, when I talk about this as I'm erasing the board, you'll recognize that when you pull those 2 strands apart and basically and unwind the helix, that you run into some physical problems of the way the unwinding is going to work. We're not going to talk about that, but there are enzymes to take care of that problem called topoisomerases that we're not going to really mention here at all. But what I do want to point out is that there are a handful of enzymes that are going to be important in DNA replication. So we'll take this section and say enzymes for DNA replication. The first one we're going to talk about is helicase. And the function of the helicase is twofold. First, it breaks hydrogen bonds. And second, unwinds the helix. The second character we're going to talk about is something known as an enzyme called DNA polymerase. The DNA polymerase is an enzyme that has 2 basic functions. First, it forms the phosphodiester bonds between adjacent deoxyribose molecules. The second thing it does is it selects the appropriate base that is complementary to the base in the old strand. Now recognize when I say that it's selecting the base, that base is going to be covalently linked to a deoxyribose molecule. But fundamentally, these are the 2 things the DNA polymerase has to do. Let me go back to this. I'm going to leave my anti-parallel figure up there. I'm going to go back to the left-hand panel here. And what I want to do on this panel is I want to imagine that we have these 2 strands of a DNA molecule. And I'm going to use some shorthand here. I'm going to use a blue line to represent the phosphodiester linkages between adjacent deoxyribose molecules. And I'm going to use a red line to represent the covalent bond between the deoxyribose molecule and its base. And then we're going to base pair these in the middle. So the blue line, and I'm going to separate this. I'm going to flare this at the end so I can put in my helicase. This is meant to be the phosphodiester linkage between individual deoxyriboses. And I'm going to say that this is the 3' end. And from what we see in the right-hand board, when we oriented the two strands in opposite directions, that means this has to be the 5' end. 3' to 5', 5' to 3'. And then to simplify things, I'm just going to put some bases in here. I'm going to choose these pretty much randomly off the top of my head. But each one of the vertical red lines is meant to represent a covalent linkage between the deoxyribose and a base. So the bases I'm going to put in here are guanine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, adenine, cytosine, guanine, adenine, thymine, cytosine, guanine, adenine, thymine. Well, I'm going to work from this end because we have these matched up. Here's a thymine. Here's an adenine. Here's an adenine. It has to base pair with a thymine. This is a guanine. It has to be to a cytosine. This cytosine has to be to a guanine. This thymine to an adenine. Adenine to thymine. Cytosine. Guanine. Thymine. Thymine. Cytosine. Guanine. Adenine. Thymine. Cytosine. Guanine, and cytosine. So we have this base pairing. So we have each base has a complement. And that complement is always adenine base pairs to thymine and cytosine base pairs to guanine. Let me change these around so it helps you keep this in order. So this will be a guanine to a cytosine. So we have the purines here and the pyrimidines here. Now the next character I'm going to put in here, and I'm going to draw it as a kind of wedge. It's not that simple. But I'm going to put this structure in here. It's kind of a wedge-shaped structure. And I'm going to label this as the helicase. The reason I shape it as a wedge is because I can easily imagine this wedge insinuating itself between the hydrogen bonds and separating the two strands by breaking the hydrogen bonds. So the helicase breaks the hydrogen bonds that link complementary bases. But remember, we said it does something else as well. It does this mechanical job of it unwinds the helix. So if you can imagine putting this in motion, what this wedge is doing is moving down the DNA molecule. And as it moves down the DNA molecule, it forces apart the hydrogen bonds, and at the same time, it unwinds that helical structure. The helical structure of DNA is an interesting characteristic. It arises because it has only the hydrogen on that number 3 carbon, and it also arises because it's the lowest order state where we can hide these hydrogen bonds between bases from water. Water has a tendency to disrupt these hydrogen bonds. So one of the ways we protect these hydrogen bonds is to throw the molecule into a helix and hide these hydrogen bonds away from the disruptive properties of the dipole moments in water. But that requires this character, the helicase, to unwind the helix. So it's unwinding the helix. And as it unwinds the helix, there's another character that comes on right behind. And this molecule, I'll put it in green, this molecule is said to be DNA polymerase. I'll remind you the two things the DNA polymerase does is it forms the phosphodiester bonds, and it also chooses the appropriate base. So it would choose the appropriate base here, a cytosine, to a guanine, form the hydrogen bonds, and then it would form a phosphodiester linkage. But this is that's not what I wanted to do. It's a form of phosphodiester linkage between that cytosine and this cytosine, because this is a guanine. So now we're going to put a cytosine here. This is a cytosine. We'll put a guanine here, form the phosphodiester linkage, put an adenine here. And then it will work its way down the line. Here's a thymine, form a phosphodiester linkage, guanine, phosphodiester linkage to a cytosine. So now what we're doing is we're inserting the appropriate base, and we're forming the phosphodiester linkages. But let me back up here just a little bit so I get the same kind of nomenclature, even though I'm a little short on room. So this is going to be the base pair of cytosine. This is guanine, adenine. These are the vertical lines connected to the backbone of the phosphodiester linkages. This goes on. Basically, this DNA polymerase gets on at the 3' end and walks from the 3' end to the 5' end, inserting the appropriate base, making the appropriate phosphodiester linkages. It's said that the DNA polymerase synthesizes, let me do it this way, reads the template strand (3' to 5', and it synthesizes the new strand (5' to 3'. So it's reading along here 3' to 5', and this end of the molecule here will be a 5' end, and we're headed toward the 3' end. Now, I think you could begin to see with this rule we've got a problem, because this strand is oriented 5' to 3'. So the simplest solution that you would think would be to simply find another DNA polymerase that could read this strand 5' to 3', attach it to this end, and read it continuously along. But that doesn't happen, because we're restricted to this rule of always reading 3' to 5'. What we have to do is we have to separate these two strands, usually a couple hundred base pairs. Separate the two strands, and someplace downstream here, we take another DNA polymerase. And this DNA polymerase reads 3' to 5', so it's going to read in that direction, but it's going to do the same thing. It's going to look at this adenine, and it's going to put in a thymine. Look at this guanine and put in a cytosine. It's going to look at the cytosine and put in a guanine. It's going to put this adenine and put in a thymine. You see that we're now making the same complementary bases. And it will form the phosphodiester linkages. So the rules are the same. In both cases, we're following along the old strand, synthesizing a new strand that's complementary to the old strand. That's the rule of semi-conservative replication. But what's different here is because of the anti-parallel organization of the two strands. The first strand we can read continuously, 3' to 5'. We can go all the way, theoretically, from one end of the chromosome down to the other, continuously reading it. But we can't do that with this strand because it's oriented in the opposite direction. So what we do is we separate the two strands over some long distance, stick the DNA polymerase on at this end, and read it in this direction. Now, for some of you who've had some biochemistry, you'll recognize I'm leaving out the idea there are primers in here. Primers are really important. They're made of RNA. We're not going to talk about them. What I want you to see is that we have these two strands, one running 3 to 5, the other running 5 to 3, and that because of that orientation, where one strand can up here be said to be the leading strand, read continuously, this is said to be the lagging strand. And it's read in fits and starts. And what I mean by fits and starts is we have to wait for the helicase to open up the strand for some distance. We put on the DNA polymerase, read it in this direction, then we wait for the helicase to open up farther down the line. Someplace down here, we put on the the DNA polymerase, read it back in that direction, and then that will form the hydrogen bonds. We again form this semi-conservative arrangement where the two old strands provide the template for the two new strands. But because of the antipolar organization of the DNA molecule, the DNA polymerase is always reading 3' to 5'. The leading strand is read continuously. The lagging strand, you read and then you wait. You read and then you wait, because we have to open up the strand and allow for the DNA polymerase toward the 3' end to settle in and read 3' to 5'. So any questions to there? If there are no questions, we can stop here, and then tomorrow we'll come around and link this idea to its cousin, ribonucleic acid.